Well, thank you very much to Korea for inviting me to give this talk today. I'm very happy to get this chance to talk to the Korean market, the Korean researchers, and introduce our technology. So what I'm going to talk about today is how you can advance your 3D cell culture using active diffusion, but at the same time don't have any shear stress. So let's just see. All right. So my name is Torsten Duebøl. I got my PhD degree from University of Southern Denmark, and today I'm responsible for the sales and the scientific support of EMEA and APAC with Celvivo. You can also find my email address here. So I'll start up by telling you a bit about Celvivo. So Celvivo was founded in 2014 by two researchers from the University of Southern Denmark, Steve and Christoph. And they were working at the university for quite some time and wanted to develop a model which had a better resemblance to in vivo than they could otherwise. And then when they worked at the university, they developed a system, and a lot of their collaborators said, okay, we would like a similar system. And then they realized that they might have something of commercial interest then. Then in 2019, they got funding from our current investors. And that meant that they could start hiring people. So I joined Celvivo in February 2020. So what they really saw was that, if you're moving from monolayers, then to 3D, you get models which mimic in vivo to a much higher degree. But that requires some equipment and some protocols. Back when Steve and Christoph were starting their work with 3D, it wasn't as popular as it is today. As you can see, the number of publications having 3D has been exponentially growing since the beginning of 2000. So Steve and Christoph, they had some requirement for an optimal research model, and I think they're quite universal and quite common. So of course, the model had to resemble reality. So it had to be a simplified model of what's going on in vivo. It had to resemble reality, have the functionality. Secondly, it had to be reproducible. You need to be able to reproduce your results, and not only yourself, but also other research groups. They wanted their model to be long-living, so they could study effects not just here and now, but also over a longer period. And finally, it should be easy to obtain these models, and there should be plenty of them. If we look at 3D cell culture, then they're divided into subpopulations. So the most popular today is called organoids, and they are normally derived from stem cells. Then you have spheroids. Spheroids are defined as 3D constructs coming from immortal cell lines. And patient-derived organoids, that's pretty obvious. It's cells from patients, and then tumoroids is patient tumors or immortal cell lines. One of the things which people normally discriminate about the spheroid and organoids is also that spheroids should be rounded, whereas organoid have crypts or could have crypts, have another architecture. Organoids are the most popular today. They are normally produced from pluripotent stem cells, so iPSCs or embryonic stem cells. So you take the embryos and then you introduce them into the derm layers, and then you differentiate and mature them into the organoids. Today, there is a numerous amount of ways to create the constructs. So you have the low binding plates, you have the transwell, the hanging drop, and the gels. And then besides that, you also have like organ on a chip, and then finally the rotary bioreactors. Where we see a challenge is that we define some of them as static, and some of those as dynamic. And with that, we mean the movement of media around the construct. One of the challenges when you have a static condition is that you very rapidly get a construct which has the size where they will use all the nutrients and all the oxygens just around them. So you get a huge depletion zone of where there's no oxygen and no nutrients, and you have a buildup of metabolic waste just around the construct, and that will suffocate the construct. So you need, if you want to have a prolonged cultivation time, you need to introduce some kind of active diffusion. You need to have the media around the construct, so you will get a constant flow of media in the preferred settings, where you get exchange of nutrients, you get exchange of oxygen, and you get metabolic waste washed away. When you introduce active diffusion using like a spinner flask or orbital shaker, you will introduce shear forces. And shear forces affects the constructs. It affects the cells, so they will be stressed, and then they will have a functionality which differs from in vivo. If you introduce a lot of shear stress, you will also maybe be able to rip the construct apart, and you will not have a uniform population, especially if you use an orbital shaker. So what you really want to do is you want to have a system where you have the benefit of the active diffusion, but you want to have the construct left in peace and quiet, having the peace to communicate with each other, create the architecture and the structure of in vivo tissue. So in the 80s, NASA actually introduced the Clinostar principle for cultivating cells. And what they did is they took a bioreactor and then they rotated it, and then the rotational force would counterbalancing gravity. So the construct within the bioreactor would be like in a static orbit, and thereby it would be like a passenger in a train carriage sitting still on its place, but the media around them would be gently exchanged. It was actually developed for simulating microgravity, but it's not really good at that, but it is very good at giving very little shear forces. So if you look at, this is our bioreactor rotating in our system. These are hepatocytes, so it's spheroids made of 52 cells. And if you try and look at one of those spheroids, you will see that they're more or less in a static orbit, only moving gently around. So they experience very, very little shear stress, but the media around them is still flowing. So we actually saw this bias of having no shear stress and active diffusion. One of the other benefits about this system is that then you actually utilize all the nutrients in your media. So our system consists of 3 parts. It consists of a Clinostar, which is our CO2 incubator. This is operated by a tablet. So the Clinostar itself is fairly small. It's 45 times 42 centimeters and then 25 centimeters in depth. So you can actually have it in your biosafety cabinet if you so desire. If we try and open the door and see inside the Clinostar, you can see it's a round. There are 6 holders for bioreactor. Each holder can be individually adjusted with regards to speed. The environment in the Clinostar can be also adjusted with regards to CO2 and temperature. And then you'll see that there are like 6 little dots in the door, and that's camera placed opposite each bioreactor. So you don't need to open the door to see what's going on in the bioreactor. You can leave the construct in peace and quiet and not disturb them too much. So they have the optimal conditions. And here you see one bioreactor. We call it a Clinoreactor. You see it all over here as well. So the Clinoreactor has its own humidification system. Each Clinoreactor has its own humidification system. And that's the blue beads you see here. So the blue beads are actually awkward beads. So the gas is flow around these blue beads, and then they get humidified. Within the bioreactor, you have an incubation chamber of 10 ml. When you place the bioreactor flat on the table, you will be able to remove the lid, and then you have it access like a petri dish. The gas exchange, I don't know if you can see it, but it happens over a membrane here, so it's a 0.2 micrometer Teflon coated membrane. Here you also see the bioreactor. You see there's a small top plug up here, and that is used for when you want to do media exchange. So it's very easy. You just simply put the bioreactor on the table, remove the stop plug, aspirate media, add new media, and then you're done. It takes like 2 minutes. One of our founders here will show you how to do a sample of a spheroid. So you see he opens up the lid and then simply remove a few spheroids, and you can also see they are fairly big. So spheroids in this system can be 2 to 3 millimeters in diameters without having a toxic necrotic core. So the benefits of our system is that you get 3D cell culture in suspension. They don't have adherence to plastic. They don't get affected by plastic. You have a unique low shear force environment. There's no depletion zones, and you can culture for a very long time. So we have cultured our HepD2 cells here, our spheroids here for 300 days. So you can culture for extreme long time. And with this, you actually get an architecture and structure which resembles in vivo functionality. So what are the benefits? Well, today we have users studying models of a wide variety of tissues. So lung, pancreatic, breasts, brain, embryos, liver, but actually also stuff like bones. But today I'm going to talk mostly about the liver, the embryo, and the brain. So here we have, again, these HepD2 cells. You see how the spheroids develop over time. So at day 3, they're fairly small, and there are a lot of them, and then they grow. And at day 29, they are fairly big, like 1 millimeter in diameter. We tried then, when they are like 3 weeks old, to do a sectioning of our spheroids and do an ECM picture of it. And if you look at this ECM picture, you will see that this has a very defined architecture and structure. We have the trademarks of a hepatocyte. We also see that they are starting to communicate with each other. So we have the bile connectors channels, we have the sinusoidal channels, and we also have these hepatocytes actually starting to have lipid droplets in here. So we see that now our cells are communicating with each other, creating an architecture and structure which resembles in vivo. And with architecture and structure comes functionality. So our cells are no longer using all their energy in proliferating. We see that the proliferation rate goes down when they're like 20 days. The doubling times is now around 20 to 30 days. But it's not because they don't use energy. We see that there's quite a lot of ATP still going on per milligrams protein. So they actually also have an energy level which resembles in vivo. But mostly important is that they actually also start behaving like liver. So they start excreting cholesterol and urea like liver is supposed to. And they do that in levels which resembles in vivo. So we try to use this model for determining LD50 values of 6 known active pharmaceutical ingredients. And if you look into the literature and see what is the prediction of these non-functional, I call them 2D cell culture, then you see the correlation between what observed in 2D and what is observed in man is 0.55. So there's very little correlation. Luckily, the pharma industry knows this, and they use mostly primary hepatocytes. And if you use primary hepatocytes, you also get a significantly better correlation of 0.75 with these 6 known compounds. However, if you take the same cheap immortal cell lines, let them grow and mature for 3 weeks in the Clinostar system, then expose them to the known APIs, then you get a correlation of 0.85. So you get a significantly better correlation than with 2D, but also a better correlation than with primary hepatocytes, simply by having a mature spheroid. And that is in itself a very nice feature. But one of the key features here is that you can grow these cells for a very long time. So that means we can also do repeated doses of the active pharmaceutical ingredients to the model. And that can help us in determining not only the acute toxicity, but also the accumulated toxicity. So here we've taken acetaminophen or APAP and treated that to our liver model. And we see if we get it at a very high dose, we see that the ATP level falls very rapidly, indicating that the cells die. However, if we halve the dose, we see here that the cells, they survive. They survive the first treatment, and we treat again after 48 hours. They also survive the second treatment. The third treatment, then we start to see a drop, but it rebounds until we give it the fourth dose, and then the spheroid, or the cells in the spheroid, dies. So here we actually have a measurement of accumulated toxicity. We also then actually went very low, so that's these 0.625. That's actually therapeutic doses. So we see that we have very little effect of the APAP within the first 3 doses, but then we actually see that the liver starts to increase its ATP levels when we give the doses, and that's simply because the liver cells are now active in trying to get rid of the APAP or metabolize the APAP. So that's also quite interesting. We are not the only one who believes that our system is good in generating models, these 3D liver models. So we have Professor Simone Sidoli here from New York, who has used the same model for studying histone modification. What Simone he also observes is the same pattern, that he gets an excretion, for instance, of albumin of the HepG2 in the spheroid, which resembles in vivo, whereas he does not in the flat culture. What Simone wants to do is he wants to see what is the immediate and long-term effect of an acetylation of liver cells. So he takes a model which has been matured for 3 weeks, and then he treats it with sodium butyrate. And what sodium butyrate is doing is it's inhibiting the deacetylation of histones. He does that for 4 days, and then he removes the sodium butyrate simply by changing media, not adding sodium butyrate. And what he sees is when he has the non-treated and the treated, he sees a huge difference in expression of biomarkers. So he gets a toxicity, or he gets an effect on the spheroids. His aim is to see if is this a reversible process, or is it a chronic process. So when leaving the spheroids for 7 days after the last treatment, he then measures again what is the expression. Is the cell recovering? And what he sees is, yes, they are recovering. This can only be done in 3D with a stable long-term culture. Because if you try to do this in 2D, most of your cells after 10 days would be daughter cells and not be the ones which was acetylated in the first place. So here you actually really utilize the liver model in 3D and the longevity as well of studying both chronic toxicity and recovery of sodium butyrate. Now I'll skip to something different, but to me it's kind of stress the same message, is that it's a very nice system for cultivating cells in suspension. So Professor Yun Wu and Carlos Pinchot from UT Southwestern Medical Center in Texas, they wanted to study blastoids from bovine. And in case you are not completely up to what blastoids is and blastocysts, then the blastocyst comes after you fertilize the egg. You get the zygote, then the embryo with the 4-cell stage, and you get the morula, and then you get the blastocyst before it evolves into an embryo. And the blastocyst has a cavity, or has a surrounding of a trophoblast, and then it has a blastocyst cavity, and within you have an inner cell mass consisting of embryonic stem cells. So what Yun Wu and Carlos did is they tried to make a blastocyst, more or less, but from iPSC and trophoblast stem cells. What they did is they took 16 iPSCs, or embryonic stem cells, and mixed with 16 trophoblast stem cells. They did that in agrowell. So they get like 16s in each microwell. They let them stand there for 4 days. As you see here, you have a picture of the blastoid in the microwell, and it resembles a blastocyst quite nicely. They looked at the blastocyst and blastoid and compared them. You see here is the microscope of the blastoids, and here is of the blastocysts. There is a very high resemblance. They measured the size, and they also did stainings, and you can see that these three biomarkers, they look very similar from the blastocyst to the blastoids. They wanted then to mature these blastoids outside the cattle, so outside the cow. So they took the blastoids and transferred into the Clinostar and let them mature in the Clinostar. And here is a small video from the supplementary of this publication, which came out last week in Stem Cell. And here you see blastoids slowly rotating in the bioreactor, and again, they can live here for quite some time. They keep them here for 14 days. Carlos is pretty sure he could keep them longer. And that's nice. They grow, so they look at the size, and you see that there's a nice growth in the blastoids, in the size of the blastoids. And then they actually take these blastoids and transfer it into a cow. And after a few days, they measure the inferior and top, and they from both blastocysts and blastoids, and they see this hormone in both cases, if the cow has gotten the blastocyst or the blastoid. So the cow actually reacts as it is is pregnant, and that's really really interesting that you can now keep blastocysts for 16 days outside the womb of a cow. And what is the perspective of this? Yeah, the perspective is, of course, immediately that you eliminate some of the bottlenecks for this material for blastocysts, which is not easy to obtain. And you can use that for studying embryogenesis and defects. On a little longer term, of course, you can look at cattle breeding technologies and study of human embryogenesis, and you can get generating a higher quality of blastocysts in vitro. And then that could maybe be something from IVF treatment in a much longer perspective. So now I talked about blastoids, talked about liver models. Now I want to talk a bit about how to use brain organoids. So brain organoids is a field which has gotten quite a lot of attention, and I want to discuss how Roman has used our system in his study for Omicron virus, COVID-2 variant. So the hypothesis of Roman's work is that variants of a virus becomes less pathogenic. What we have seen is that, or what is seen is that Omicron virus manifests itself in the neurological pathway, and there's different variants within the Omicron virus. So the BA1 should be less pathogenic than BA5. So BA5 is actually more pathogenic than its ancestor. That's the hypothesis. So what the group in Australia did was they infected mice with BA5, and they saw that it was indeed infected in the cortex, in the hypothalamus, and in the hippocampus of the mice. So then they saw if they were actually also located in the neurons, and doing these stainings with both neurons and BA5, they saw that they were indeed in mice affected, and the BA5 was more pathogenic in mice than the BA1. But mice is not an ideal model for humans, so the next step was to generate a cortical brain organoid to study this well. So when you do a brain organoid, what you do is you make an aggregation, make an embryonic body, and then you treat that with different growth factors, so you get the neural induction, then you do the expansion and the differentiation and the maturation. And that was exactly what Roman also did. So he did the embryonic body in ULA plates. You see up here. He then did the induction also in the plates. He embedded them actually in Matrigel, and after day 10, he transferred the constructs into the Klein reactor and did the differentiation and the maturation in the Klein reactor. He did that for 30 days. And if you look at his brain organoids over here, he has 24 brain organoids in one Klein reactor. You see they're very uniform, and they're also fairly big. They're quite big, so it's 2 to 3 millimeters in diameters. And the uniformity is extremely important if you want to compare BA5 and BA1, because you need to have kind of the same benchmark. And that was what they did. They tried to take infect BA5 and BA1 with the virus, the brain, the BA5 and BA1 virus to the brain, and they saw that a larger proportion of the cortical brain organoids is actually infected with BA5 and hereby display a higher pathogenesis. So here the conclusion is that BA5 is actually a more pathogenic variant than its ancestor. They also looked at the titer and saw that BA5 does actually give a few more virus particles than the BA1. So that was So Rommel used this Omicron virus, or this brain organoids, to study Omicron virus. But actually here 3 weeks ago, he then also used the same model now for studying this Japanese encephalitis virus, and looking into which virus variants are the most infective and most harmful ones. And again, he could use the same brain organoids, and he could then actually, again with mouse data, correlate that indeed the Nakamaya variant was more pathogenic than the other variants. If you want to know a bit more about brain organoids and neural organoids, you can download a white paper we have made here at our website. So I just talked about a lot about brain organoids, talked about blastoids, talked about liver models, but we also see quite a number of users using it for studying variant cancer types. So that is small lung cancer, small cell lung cancer, colorectal cancer, liver cancer, thyroid cancer, and glioblastoma, and so on. So you can use it for a wide variety of cell lines as well, of course. You can actually also do co-cultures. So I started out saying that we wanted a model which resembled reality. It should be reproducible, it should be long-living, and it should be abundant and easy to obtain. I think I've shown some examples of this, but I also just wanted to make you aware that it's not only me who's saying this. So Krishna here, she's saying that she obtained physical relevance. So I think kind of goes with the functionality or the relevance. We have Jay here, who's actually also doing brain organoids in our system in a different manner. And what he really likes about it is the reproducibility, the ease and to operate, and how they get abundant material. We have Christoph Deben here, who's using it for doing some very exciting cancer studies, hoping he will publish very soon, but they really utilize the long-term cultures of our system. And then we have also Markus here from Germany, who are also using it for these long-term studies. So we have quite a number of users who's actually can recognize that they get the benefits which we will set out to have in our system. So we also have neurospheres and other studies. Yeah. Good. So to sum up, the Clinostar system allows you to make reproducible experiments, get organoids or spheroids in abundance. It's fairly easy to operate, and it actually creates models which has a functionality which resembles in vivo. Then I would like to thank FileKorea again for giving me this opportunity. So FileKorea is our exclusive partner here in Korea, and a very trusted partner. We are very happy with how Korea or File Korea is assisting our customers. So thanks again.